So as it's filling up, it's a pleasure to replace Eli Sprecher and Bernhard Home, who unfortunately cannot join today. And I have the honor and the pleasure to chair this session, this week's ESDR Kitchen Live webinar. It's a molecular cuisine. We have Professor Michel Gilliet here. He doesn't need much introduction, actually. He also asked me to keep it short. He's actually sitting in the next room. So Professor Michel Gilliet is a full professor and the chair of dermatology here in the Department of Dermatology in Lausanne, Switzerland. He asked me to keep it short, so I do that. He's a former president of the ESDR. He has published numerous publications in absolutely outstanding journals such as Nature, Nature Immunology, Nature Medicine, and so on. Has more than 25,000 citations, and he's going to talk about how he kept chasing triggers of early inflammation. So I hope that was short enough. Michel, the stage is yours.
Thank you very much, Curdin, for the introduction. So I will be talking about what are the triggers of sterile skin inflammation. This was a question that haunted me for the past 20-25 years since I started my derm residency and my research career. At that time, a lot was known about infectious inflammation with the discovery of toll-like receptors, but the initial triggers of sterile skin inflammation that occurs in diseases such as psoriasis and lupus, but also in skin wounds, was not known. And at that time, I chose to do a postdoctoral fellowship in the lab of Yanjun Liu, who just discovered at the beginning of the 2000s plasmacytoid dendritic cells. This is a very interesting subset of the dendritic cell that expresses selectively two types of toll-like receptors, which are toll-like receptors 7 and 9. And these receptors are located in endosomal compartments, and they allow plasmacytoid dendritic cells to sense viral infections. So RNA virus or DNA virus can respectively activate toll-like receptor in toll-like 7 and toll-like receptor 9. On the other hand, DNA and RNA, which is extracellular, cannot access this compartment, does not activate these cells. And in response to toll-like receptor 7 and 9, PDC, so plasmacytoid dendritic cells, I will call them from now on PDC, they produce huge amount of type 1 interferons. It has been shown that about 70% of the transcriptomic of activated PDC are dedicated to type 1 interferons with production of interferon alpha, interferon beta, and interferon omega. So upon activation and production of type 1 interferons, PDC play a key role in antiviral immunity. On one hand, type 1 interferons blocks viral replication and entry into cells. And on the other hand, it shapes antiviral immunity. It activates conventional dendritic cells. It promotes antigen presentation, so viral antigen presentation, cross-presentation to CD4 and CD8 T cells. So a key role in antiviral immunity. But coming back from my postdoc into dermatology, I asked myself, do PDC exist in the skin? In normal skin, you cannot find them. But that was the time at that time, so beginning of 2001, 2002, Aldara cream, which you all know, this is topical imiquimod, came onto the market. Aldara is a TLR7 agonist, so I asked myself whether PDC would be present in imiquimod, so Aldara cream treated skin. So we did two studies here, so on basal cell carcinoma, and indeed, when you treat this basal cell carcinoma, we find large amounts of PDCs infiltrating, so BDCA2 is a marker for PDC infiltrating the tumors. And very interestingly, we treated here a patient, a psoriasis patient, for its bounce disease with Aldara, and what we found is a very large accumulation of PDCs and exacerbation of this psoriasis. So we wanted to look a little closer on what is going on here in psoriasis. So we extended our studies to psoriatic samples independently of imiquimod treatment. And interestingly, we did not find any PDCs or very low PDCs in chronic plaque psoriasis, but we found quite some amount of PDC infiltrating early developing psoriatic skin lesions. So we then looked and used the xenograft model where we transplant uninvolved skin of a psoriatic patient onto an immunosuppressed mouse. And we know that this transplant then develops fully fledged psoriasis. We looked into this graft and we saw that indeed there is a very quick activation of PDC to produce type 1 interferon, leading then to a Th17 response and development of psoriasis. And we showed in this model that if we block interferon receptor signaling or we block the ability of PDC to produce interferons, we can completely block the development of psoriasis. So this indicated to us that PDC's infiltration activation to produce type 1 interferon is a very early trigger of psoriasis pathogenesis with the activation of conventional dendritic cells, Th17 cells, which by producing IL-17A, F, IL-22 induce the psoriatic phenotype. And so the question, of course, was what activates this PDC in psoriatic skin? And we collaborated with Jens Schröder from Kiel. He gave us reverse phase HPLC fractions from psoriatic scales, and we used these fractions to stimulate PDCs. And to make a long story short, we identified some of these fractions were able to activate PDCs, and this fraction contained antimicrobial peptides. One of these antimicrobial peptides, which I will be, which we're studying more, and I will be showing some data on that, was LL37. So LL37 is an antimicrobial peptide, 37 amino acids. It's highly cationic, but amphipathic, meaning that on one side of the molecule we have a cationic, cationic residues. On the other side, we have it's more hydrophobic, and these unique structures allows them to associate with bacteria and form pores in them and killing them. So we looked at L37 expression in psoriasis. It's expressed throughout the entire epidermis. We know that expression is not present of L37, not present in healthy skin, but it can be used in skin wounds, and it's highly overexpressed in psoriasis, as shown here. The source is keratinocytes, but also neutrophils. Infiltrating neutrophils can release preformed LL37. So this does not explain how PDCs are activated. As I told you, these cells uniquely have toll-like receptors 7 and 9, which sends RNA and DNA in endosomal compartments. So with reasons that this cationic peptide could maybe bind extracellular self-DNA, so human DNA released by dying cells, and by forming these complexes, allowing them to enter these endosomal compartments and triggering toll-like receptor 9 in PDC. And this was indeed the case. So human DNA, which is usually soluble, when you combine it with L37, it falls out of solution. We can readily stain it on the bottom of the well. And when we give these complexes, so self-DNA with L37 to PDC, we see that the DNA is able to enter endosomal compartments and it co-localizes with TLR9. More importantly, these PDCs are strongly activated by these complexes through TLR9, while human DNA fragments, so self-DNA fragments given to PDC, because they are not able to enter the cells themselves and they're rapidly degraded, they are unable to activate PDCs. So this again is a schematic of the mechanism. We have self-DNA fragments that are released by dying cells, for example. These can complex with antimicrobial peptides, such as L37, others can also do it. These complexes are slightly positively charged. They associate with proteoglycans on the surface of the PDCs and enter endosomal compartment where they trigger toll-like receptor 9, giving rise to high levels of interferon alpha and beta production. Another key mechanism which we discovered a little bit later is that this complex formation induces a very regular structure in which the DNA molecules are spaced among each other with a space of 3 to 4 nanometers. And this allows them, and this is shown here, to interdigitate perfectly TLR9 and use high levels of type 1 interference. In this study, we showed that complexes which space the DNA at less than 3 nanometers or greater than 4 nanometers are unable to interdigitate the TLR9, and therefore they're not able to induce efficiently type 1 interference. So coming back to the pathogenesis of psoriasis, this is what we propose, is that mechanical stress induces some antimicrobial peptide expression, L37. which forms complexes with the extracellular DNA originating from dying cells. These complexes are able to enter intracellular compartments of PDC, leading to the production of type 1 interferons, which ultimately elicits the downstream inflammatory cascade, which triggers psoriasis with activation of conventional cells, dendritic cells, Th17 cells, which ultimately are responsible for the induction of the psoriatic phenotype. So in the years after this initial finding in psoriasis, we found that there are also other receptors and other cells which can be activated by these complexes. So the DNA L37 complexes, they can also gain access to cytosolic compartments of macrophages and monocytes, which the human ones do not have toll-like receptor 9, but they have this cytosolic DNA sensor, which is cGAS-STING. And upon activation by the DNA brought into this compartment by the L37, they also produce type 1 interferons. And we also found that RNA molecules can bind with L37 and induce pro-inflammatory cytokines in conventional dendritic cells. So one of the questions at this point was whether these complexes can also elicit adaptive immunity. I've shown you for viruses that type 1 interferons critically shapes downstream dendritic cell activation, conventional dendritic cell activation, and antigen presentation. So we asked here whether these complexes, and in particular the L37 part, could be activating specifically CD8 and CD4 T cells in psoriasis, and therefore function as a T cell off antigen in psoriasis. And this was indeed the case. So when we take psoriatic patients, PBMCs, and stimulate them with L37 compared to a scrambled form of the peptide, we see proliferation of T cells, and these T cells were poised to produce IL-17A, so they were of the Th17 phenotype. And what we also found is that the decrease of the PASI upon treatment, shown here from 20 to 5, directly correlated with the decrease of L30 specific T cells, shown here by T cell proliferation to L37, and here directly by tetramer staining, which indicated that these L37 specific T cells, they could be pathogenic of psoriasis. Indeed, they correlate with disease severity. We found them in lesional skin, and they produce Th17 cytokines, which are pathogenic in psoriasis. So this is the model we proposed. L37 is produced upon mechanical stress, formation of complexes which activate PDC interferons. Some of these RNA complexes can activate directly the conventional dendritic cells, and these cells then present efficiently L37 to autoreactive Th17 cells, which migrate into the epidermis. The keratinocytes also produce L37, express L37, so they present L37 to mainly CD8 T cells, which then produce Th17 cytokines, inducing the psoriatic phenotype. But importantly, these psoriatic cytokines, so IL-17A, F, and IL-22, they further increase the production of L37. This has been shown by other groups, and therefore they amplify these the L37 production, and they amplify PDC activation and the inflammatory loop in psoriasis. So a second disease which has PDCs is lupus. So we stained cutaneous lupus for PDC, and we see quite some numbers of PDC infiltrating these lesions. So PDC are known to be activated by immune complexes in lupus-producing interferon alpha, and this then elicits the downstream inflammatory cascade. However, how these immune complexes, which contain DNA, activate PDC wasn't actually not known. And what we looked is whether these immune complexes also contain these antimicrobial peptides, such as L37. This was indeed the case. Here is simple staining of immune complexes that fall out of solution for DNA in L37. And we found that these complexes originate from neutrophils. They contain also other neutrophil-derived proteins. They originate from neutrophils, which have here in orange L37, and they are able to release it in the context of neutrophil extracellular trap formation, where they extrude very large amounts of DNA coupled with L37. And these complexes are then able to activate PDC to produce type 1 interferons. And they are at the origin of the immune complexes. So what we also found in lupus is that there are T cells specific for IL-37, just as in psoriasis. So here we stimulated PBMC from SLE patient or cutaneous lupus patients with IL-37 or control peptides, and we find high levels of proliferation. However, in contrast to psoriatic patients, these T cells had the phenotype of follicular T helper cells, which are the T cells which promote B cell activation and antibody production. In fact, these T cells produced IL-21 and less IL-17. They express PD-1, ICOS, and CXCR5, which are markers for follicular T helper cells. Furthermore, we find high levels of anti-IL-37 autoantibody production in lupus, shown here, compared to other diseases such as scleroderma and healthy donors. And when we stimulate, when we use this anti-IL-37 antibody to stimulate neutrophils, we found that anti-IL-37 antibodies directly bound to IL-37 expressing neutrophils and induce the formation of nets, which, as I've shown you, contain these IL-37 DNA complexes. Therefore, we propose the following model. We have activation of neutrophils to release nets, formation of DNA IL-37 complexes, which activate PDC produce type 1 interferons, and this then elicits follicular T helper cells and B cells to produce anti-IL-37 antibodies, which further amplify neutrophil activation with the release of more complexes that activate PDC, again providing a feedback loop which amplifies autoimmune inflammation. So a third condition in which we can find PDCs in the skin are skin wounds. Here I show you skin wounds. We have upon wounding, we have a very rapid PDC infiltration, which, however, in contrast to psoriasis, lupus is very transient. We found the mechanism. We know that the microbiota, which infiltrates the wound bed, stimulates antimicrobial peptide production, and this leads to the formation of DNA complexes, which then enter PDC and activate PDC to produce a transient interferon production, which is accelerating wound healing through the induction of VGF and FGF in fibroblasts and macrophages. So basically, we have a physiological function of this PDC interferon alpha beta production. So how do we put all this data together? We think we have this physiological function in the context of wound healing. However, in psoriasis, this activation leads to an overactivation of Th17 cells producing IL-17, which then activates keratinocytes, produces further antimicrobial peptides, further complexes, and provides a feedback loop which sustains this inflammatory process leading to autoimmunity. On the other hand, in lupus, we also have type 1 interferon induction by PDC, but this rather induces activation of follicular T helper cell, and T cell activation with production of autoantibodies which further stimulate neutrophils to produce complexes which activate PDC. Again, self-sustaining inflammatory feedback loop which drives autoimmunity. So why we have these two conditions? I think the increased Th17 induction by PDC and interferon is due to genetic polymorphism. Indeed, there are several genetic polymorphisms that have been found associated with psoriasis, some of them clearly associated with enhanced Th17 response, such as IL-23A and IL-23 receptor. On the other hand, the genetic polymorphism associated with lupus, they are linked to B cell activation and autoantibody production. And so we think that this physiological activation of PDC interferon triggers a B cell response, which then specifically provides a feedback loop, further activates neutrophils, and further activates PDC, providing this feedback loop and for the development of autoimmunity. So these were two cases, but I think there's now more and more evidence that these IL-37 DNA or AMP DNA complexes and PDC interferon activation drives a lot of other inflammatory diseases. We have shown psoriasis, lupus. Roberto has shown also scleroderma may be driven by this pathway, and several groups. I'm just mentioning one of the references, but there are several and this is well established, have shown that this pathway also drives type 1 diabetes and also atherosclerosis and small vessel vasculitis. So it is really a pathway which is plays a key role, and this aberrant self-DNA sensing is a key role in many chronic inflammatory diseases. What also has been found in the past years is that L37 is of course not the only antimicrobial peptide, or not the only peptide which can break innate tolerance to DNA, which can bind the self-DNA, form these immunogenic complexes. We know that other antimicrobial peptides can also do it, beta defensin 2, 3, lysozyme, RNase 7. But more recently, we and others have found that chemokines, they also have this unique structural feature with cationicidity and amphipasticity, and they are able to bind DNA and promote the self-DNA sensing. So 6L9, 10, 11, 6L4, 6L12, and 6L5. We were very interested in a cytokine because we identified that there is a cytokine, IL-26, produced by Th17, which has also this structure and is also able to kill bacteria, so has an antimicrobial activity, and importantly also drives self-DNA sensing by forming complexes with self-DNA. This is very interesting because IL-26 has also its own receptor, so we think a combination of these different effects is very interesting and very important in several chronic inflammatory diseases. And I think this is very exciting. I think we started from the skin, from psoriasis, went to lupus, to skin wounding, and we identified really a key inflammatory pathway based on the aberrant sensing of self-DNA and PDC activation, which plays an increasing role in several chronic inflammatory disease as initiator. And I think this pathway needs to be targeted therapeutically, and we have already some exciting possibilities now. So we can block the interferon signaling through anifrolumab, which has been accepted by the FDA for lupus. We have the TYK2 inhibitors, which inhibit IFNAR signaling. We have PDC inhibition. These are tested in clinical trial, anti-BDCA2, anti-IL-7. But I think the future will also be TLR and STING antagonists, so we can block the receptor. We need to target nucleic acid. We can target IMPs, and we have generated a humanized antibody, which we are now pushing for clinical development that targets IL-26, which we think is a very important antimicrobial peptide driving inflammation, several diseases. And with that, I come to the conclusion of my talk. I would like to give not a scientific conclusion, but a conclusion from, as I call my kitchen. I think it's more for the younger generation. I think what we have been doing, and what I think is important, that you develop your signature dish, so your research line, and you follow a thread over the years to try to prove or to discover things along what you are researching. Also, it's very important that you combine multiple ingredients, so both take advantage of your clinical expertise, but combine with translational research and go to the mechanism by using basic research. And I think ultimately you need to think broader about the broader impact of your research. Skin can be a model for many biological processes aside from the skin. And of course, very exciting is the possibility to discover new therapeutic targets. And with that, I would like to, of course, thank the cooks in my lab over the years. So here is a big list with senior scientist, postdoctoral fellow. You see also the chairman was many years ago, was with me, the PhD students and the lab technicians, and of course the generous fundings over the year. And of course my department in Lausanne, and the key collaborators, you see them here. I don't have time to list them, but I also would like to thank you, of course, for your attention. And I'm happy to take questions. If you don't have time to take questions, I can answer some of the questions also by email. Thank you very much.
Thanks a lot, Michel, for this very clear and excellent presentation. A lot of data, a lot of concepts also in a short time. I repeat it here, you can put your question into the chat or the Q&A section, and we'll discuss it with Michel live. The session will also be put onto YouTube probably next week, so you can have a look at it again if you missed some parts of it. So we already have one question from Vijay Patra asking if, first of all, very nice presentation as well, and asking is LR37 still expressed in healed lesions of psoriasis patients?
It is a good question. I have to admit that I haven't looked specifically, but I would rather say no, it's not expressed. I mean, we know that in non-lesional skin of psoriatic patients just outside the plaque, it's not expressed anymore. So I would say it's not expressed anymore. It's a completely healed lesion.
Do you think there might be antimicrobial peptides which are just on a very low level constitutively expressed to control also bacteria that we have on our skin, like the microbiome?
So it's also a very good question. I think we looked at that quite extensively. I mean, there are some antimicrobial peptides which are expressed constitutively in the skin, and also they are also expressed in non-lesional psoriasis. But these antimicrobial peptides, which have an antimicrobial function, they do not bind DNA, and so they do not activate PDCs. So I think it's these DNA binding antimicrobial peptides and PDC activated antimicrobial peptides are really only there in overt inflammation, and not. But it's an interesting thing to look at in more detail. I mean, I don't have the ultimate response because I haven't done the experiments.
Maybe you showed that. I'm just asking. We're looking for several additional questions, but you showed that you say the downstream of interferon is mainly driven by the genetics or decided by the genetics. Do you think that it could also be that the upstream is somehow different, that you have maybe, I don't know, and different antimicrobial peptides which are distinctively expressed and different bacteria or so that then drive a different response downstream?
Yeah, absolutely. We don't have clear evidence for that. So far it looks like this is a physiological pathway which then is downstream. You have a sustained autoimmunity which feeds back to the upstream, right? But people have looked at, for example, if they find copy number polymorphism in certain antimicrobial peptides, and there seems in psoriasis they have found it, but I'm not sure whether these data are really very solid and have been confirmed. But it's absolutely possible that it could also be linked to the upstream, right?
Yeah. And maybe also a bit of hooking up on that. You haven't mentioned viruses, so do you see endogenous viruses playing a role in shaping the immune response? There's like
yeah, it's also very good. Very good question. I mean, I'm sure you know about Yasmin Belkade, who has shown basically that this interferon response, which we see in the skin, she sees a low level. We see after wounding, she sees a low level at all times. She thinks they are driven by reactivation of endogenous viruses through skin. So it could be that this plays an additional role to the self-DNA driven pathway, which we have uncovered. Yes.
And then maybe the last question from me, but also it's always going a bit in the same direction. What is the role of the keratinocytes and other sources? So different type 1 interferons? Are they just bystander, and interferon is the early, driver, and then keratinocytes or other cells take over, or is this dependent on the disease?
So Richard Galla has shown that basically these complexes and the L37 can also activate keratinocytes to produce some probably not interferon alpha, but more interferon beta or other interferons. So we showed that this plays a role also in wounding and psoriasis. In our experiments, and I'm sure you, Curdin, you were part of that. It's very difficult to activate keratinocytes. That, I mean, it can happen. Probably they take up these complexes, but we think the immune system is 10 to 100-fold more potent in inducing these interferons than keratinocytes themselves. It's still possible that there is endogenous mechanism that activate these keratinocytes, and this is probably the case. So endogenous virus, as you just mentioned before, but I think for the DNA from extracellularly, the main targets are immune cells. They produce much more interferons than keratinocytes.
Thanks a lot. We're not having any additional questions. It's obviously that clear that no one has any questions additionally, or they're all writing some emails to you privately afterwards. So thanks a lot, Michel, for this really great presentation again. This was the last ESR Kitchen webinar of this year. The next one's going to be on 18th of January. Yeah, I'm not saying any wrong data. So it's 18th of January. It's going to be a freshly baked. The 2 papers will be announced, I assume, in the coming weeks. Please don't forget also, this I already mentioned, the webinar is going to be on YouTube soon. And don't forget, on January 10th, so in about approximately 3 weeks, there's a deadline for the abstract submission for the ISID in Tokyo this year. It's going to be in May. And we also have close to 200 travel grants available. So also for the young colleagues, please apply for that. There are several countries have applied, several companies have also supported this. So please submit also your applications for that. And with that, I'd like to thank you all for your attention, and Michel again for your excellent presentation. Thanks a lot, and have a happy holiday. Bye-bye.